Welcome back. In this video, we'll continue our journey to put everything together in TEM. In the previous video, we looked at the illumination system, that's from the electron source to the upper side of the specimen. We're now in a good position to focus on the objective system and the imaging system. We start from the bottom surface of the specimen to where the electrons are getting detected. There are three types of information we try to collect. These are diffraction patterns, TEM images, and STEM images. Let's look at the formation of diffraction patterns first. You should be familiar with what we are looking at now. So we have the specimen here, the objective lens, the intermediate lens, and the projector lens. In the diffraction mode, what the TEM does is it projects diffraction pattern on the viewing screen. You may notice the diffraction patterns are formed way before they reach the screen. You see them here at the back focal plane. Then the image of the diffraction pattern formed again after the intermediate lens and magnified by the projector lens and captured on the screen. When you switch from the diffraction mode to the imaging mode, the ray diagram above the intermediate lens does not change. It's exactly the same as the one from the diffraction mode. However, after the intermediate lens, because the lens strength increases, it bends the beam a lot more. So instead of forming the projection of the diffraction patterns, it forms the projection of the actual image. One interesting thing is that even you're in the imaging mode, you still form diffraction patterns. And it's right here. We're just not projecting these diffraction patterns on the screen anymore. In terms of the TEM operation, when you're in the diffraction mode, you retract the objective aperture and insert the SA aperture. In the imaging mode, in most of the cases, you insert the objective aperture and you always retract the SA aperture. In the imaging mode, you can acquire bright field images as well as the dark field images. I found a good example online. The paper was published by Xu and co-authors in the journal called Optics Express. It is a multi-layer thin film specimen, consists of the yttrium layers and the palladium layers. The figure on the left is a standard bright field TEM image. On the right is a dark field image. What the authors did was they selected the yttrium spot in the diffraction pattern to form the image. From the dark field TEM image, you can see the yttrium layers, they just light up. How can we form the bright field and the dark field TEM images? Let's look at these three examples. I also made some animations here. The red cross is the optical axis of the instrument. The large green spot is the diffraction pattern from the direct beam. The small spots are the diffraction patterns from the diffracted beam. Assuming we are tilting the specimen to a low index zone axis, and assuming that you have done a great job with the instrument alignment, the diffraction spot from the direct beam should coincide with the optical axis. To form a bright field image, all we have to do is to insert the objective aperture to only allow the direct beam to form the image. What if we want to form a dark field image using one of the diffracted beam? The layman's approach is to insert the objective aperture and form the image only using that diffracted spot. However, this approach doesn't give you the optimum results. The reason is because that for the instrument alignment, we align the beam along the optical axis. In this case, the image formed is from the diffracted beam, and the diffracted beam is not aligned along the optical axis. I call this approach the layman's dark field. The proper way is to perform a function called the dark tilt inside TEM. Essentially, you tilt the diffracted beam along the optical axis. Then you insert the objective aperture to form the image. The last type of the imaging formation is STEM. STEM essentially can be viewed as SEM. The difference lies in how the information is collected. In SEM, the secondary electron detector as well as the backscattered detector are mounted above the specimen. In STEM, the scanning transmission electron microscopy, the detectors are mounted underneath the specimen. The example here is taken straight from the textbook. and the specimen is the gold nanoparticles supported on the carbon film. In STEM, there are two types of detectors. We have the bright field detector as well as the annular dark field detector. Depending on the collection angle, the annular dark field detector can be high angle annular dark field detector called HARDIF. If you recall what we learned in the elastic scattering, higher Z elements are more likely to scatter at larger angles, so the HARDIF detector gives us the Z contrast. So the gold nanoparticles will appear bright in the HARDIF STEM image. The bright field STEM image is very similar to the bright field TEM image. There are some minute differences in terms of the contrast we see. We'll discuss more in the third part of this class. This is the very last part for the instrumentation of TEM. In the next video, we'll discuss sample preparation for TEM.